The current video is designed to teach you how to organize your files relevant to Stata on your computer. When you start out with Stata, you actually are starting with files that are going to input into Stata, which are not Stata format. And so I call these source files. And these source files generally come in several different formats, probably the most common being text files, CSV files, or Excel files. Of course, you can't use these in Stata, and so you want to run a series of commands which you'll encode in a do file, which you haven't learned about yet, you will, which then take these files into what I call generated files. These generated files all have the same suffix indicating that they're a Stata file. They're all called DTA files. Of course, you're going to have several of these, each corresponding to different streams of data from different parts of the study. And you're going to want to now merge together these streams of data and put it together into something which you can actually turn into some sort of meaningful results. Or in our case, we like to group them into things that will actually turn into a manuscript. This includes all the results of the manuscript, all the figures of the manuscript, and then all the data you're going to need for any of your tables. There's also a do file, which is used to transform these generated files into a single manuscript file. And each do file of this sort should lead to a single manuscript. So how do you distinguish these two different types of do files? Well, you want to actually label them in a very consistent way. So each of these files will be labeled as such. So these do files will be labeled in three parts. The first part will indicate the study, and then I put an underscore afterwards. Then afterwards, to indicate that the purpose of this file is to prepare and clean the source data, I use the nomenclature prep clean. And then finally, it's not going to prepare and clean all of the data, but just data from a single stream of the data. And so the last part is the stream of data, for example, demographics, cataracts, etc. This do file will be a little bit different. So it's going to now also refer to a specific study, but it's going to do the statistical analysis for a study, and then it's going to produce a paper of a single purpose. And so it'll have a three-part name again, and separated by two underscores. It was just study, stats, and paper. So let's actually show you how this works in all my files. I should also mention that the generated files also have a very specific name formatting. And so these are going to be labeled according to their study and the stream of data. Whereas these will also be labeled probably however the database manager has designed it. So let's show you how this actually works in practice. So these are all the different projects that I'm working on right now. In each of them, there's some analysis that needs to be done in order to produce the papers that we're going to generate. We'll talk specifically about a single project called Reading in Glaucoma and Dry Eye. Within that, you'll see several folders that relate to the IRB, the protocols and procedures, writing the manuscripts themselves. But before you can do that, you actually want to do the analysis that's going to produce these manuscripts. It's very important that this folder is labeled exactly as such, and you'll learn about why that is later on. But basically, a very precise naming and consistent naming system allows you to share files with other people in a way that doesn't create confusion. So I always name it analysis, and I always have a capital A at the beginning. Once you go into the analysis folder, you'll see several different folders within it, as well as several do files outside of it. So the first do file folder that I want, or the first folder that I want you to pay attention to, is the source folder. Like I mentioned before, source files are often TXT files, and you'll see that there's a whole slew of TXT files that relate to different streams of data. For example, cataract, chart data, demographics, medical history, etc. Of course, you want to actually convert those source files into something that's usable by Stata, and so you'll see just what I described earlier, that there'll be several folders that are labeled. There are several files that are labeled read to indicate the study name, prep clean to indicate the purpose of the do file, and then finally describe which stream of data they prepare and clean. For example, the cataract, the comprehension, the demographics, the medical history, etc. Once these prep clean files are run, you actually generate a file which is a DTA file, which you'll now find in the generated folder. For example, here, as I mentioned, is also labeled read for the study name, and then it's labeled by the stream of data, and then it has a DTA suffix to indicate that it's a Stata file. And again, you'll see a chart file, a cataract file, a comprehension file, a demographics file, etc. Finally, you want to take all of these DTA files and you want to do an analysis on them so you can actually get results that will provide you the information that you need for a paper. So this shows one example of that. So then again, this overall study name is read, and the purpose of this do file is to do the statistics, and it's going to do the statistics to analyze the reading questionnaire. And so you see that right over here, and you have a do file which is meant to do the statistical analysis on the reading questionnaire. We'll go into each of these do files and show examples of them and specifics of them in more detail afterwards, but this is a good start. Thank you.